This week, Dr. Rahal is joined by Patricia Zilliox, who had an extensive career with Alcon before joining the Foundation Fighting Blindness and then Eyevensys. Patricia shares her path that led her to her current role as the CEO of a biotech startup developing a gene therapy to treat uveitis. Let's listen in.
Welcome back, everyone. This is, again, the OIS Retina Podcast. This is Firas Rahal. I'm the host, and I'm a partner at Retina Vitreous Associates here in Los Angeles, and I'm also a partner at Excite Ventures, a small venture capital firm that invests essentially strictly in ophthalmology based in New York. I'm delighted to have today as our next guest and our newest guest, Patricia Zilliox. I've known Patricia because we've talked before about her company and my professional capacity through Excite, and they've made a lot of progress. And so I thought it'd be great to have her on and to update us about Eyevensys. She's the current CEO of Eyevensys. That's spelled Eyevensys for those of you who are not sure, because it's a tricky word to spell. Patricia has had many positions before in the ophthalmology world. Many of you know her because of her stint at the Foundation Fighting Blindness. I'll give her a chance to tell us a little bit about that. She was Chief Drug Development Officer at the FFB and was also at Alcon for many, many years. It looks like, Patricia, it was about 20 years. You can correct me if I'm wrong, at Alcon, also in the drug development realm. Why don't you tell us a little bit about your background and essentially how that brought you to this current position at Eyevensys?
Sure. So yes, indeed, I spent 25 years with Alcon here in Texas, because I'm actually based in Texas. So I left Alcon in 2011. I was head of clinical ophthalmology, but all my years at Alcon, I focused mainly on retina, on clinical development in retina. So I was trained by many of you, like David Bonheur, especially David Geyer, Jason Slachter. All your colleagues, they taught me what the retina was in the eye and how to read an angiogram and how to read an OCT. That's how I started in retina. When I left, and I spent my 25 years with Alcon, I left in 2011. And at that time, I joined Foundation Fighting Blindness in Washington, D.C. And my role over there was really, the foundation is trying to advance the development of treatment for treatment of disease of the retina due to gene mutations. And my role was not to give grants away. My role was really to work closely with the FDA and the NIH to try to identify new clinical endpoints so we can help companies to develop treatment, because clinical endpoint is key in this disease, which are very slow progressing. And at the same time, I was also doing seed money, not like a venture capital, but doing seed money to create new companies, to try to advance them again in the field to get treatment to the patient with retinal disease due to gene mutation. While I was doing that, we created with Professor Sahel in Paris, who's in Pittsburgh now, we created a company, Sparing Vision. It was gene therapy. And while I was over there, I met BPI, a French investment group, who introduced me to Eyevensys. Eyevensys was a company which was created by Professor B.R. Cohen, based in Paris as well. And their approach was really to try to develop treatment for disease of the retina as well, but not using gene therapy. And to make my story short, for personal reason, I had to leave, for family reason, I had to go back closer to France at that time. And Eyevensys, the board, the investor, offered me to take the job as CEO. That was not really what I wanted, because I was very happy with Foundation Fighting Blindness. But being French, living in the United States, coming from ophthalmology and personal family reason, I decided to take the job. So I took the job in the end of 2017. I really liked the technology. I loved it. But for me, there were some requirements before I took the position, was really to bring the technology back in the States, because I knew that we needed to do this clinical trial and do evaluate this technology here in the United States. Otherwise, it would not be accepted in the field. And that's what we did. We incorporated the company last year in the United States, in Dallas, obviously, because I'm still based here where Alcon is. We incorporated the company. We filed an IND for one of the assets. And we launched the clinical trials here at the Cleveland Clinic in Cleveland. At the Cleveland Clinic in Cleveland, right? Correct. And we also secure Series B fundraising of 30 million euros. That's a summary, high level.
Great summary. And we definitely are going to get into more about what the company's doing right now and a little bit about the science. I know it's a little complicated, so I won't go into the greatest detail so our listeners aren't mesmerized. But tell me, I know that the company itself, it's generally speaking, an ophthalmology gene therapy company, but it has this very unique delivery method, which I think is of the greatest interest for us to hear about. And I know your initial indication or target is posterior uveitis. Can you tell us a little bit about the history of the company, how this came to exist as a concept, and then maybe a little bit about this technique of non-viral vector gene therapy?
So really the idea, all the credit goes to Francine, of course, Francine Béard-Cohen, who is the founder. And her idea was really to try to get treatment to the back of the eye. Avoiding the virus, gene therapy, it's very sexy, we all agree on. It's great for to correct gene mutation in case of RP, but there are a lot of issues associated with gene therapy, especially if you treat AMD, for example. So she was trying to avoid the virus. She was trying to create a different approach to produce a protein in the back of the eye. So that was really her idea. And the way she approached it, she looked at the eye. She said there's a ciliary muscle which is available, which is basically there, and which could be used. So the idea was to take plasmid, to encode them to produce therapeutic protein anytime. That's pretty easy to do. Manufacture of plasmid is very easy to do. But instead of putting them in a virus, is to inject them in the ciliary muscles. Well, if you just do that, nothing is going to happen. So you need to do electrotransfection. Electrotransfection is the whole technique. All what it does, it allows the plasmid to penetrate in the ciliary muscle cells, and then the cell machinery takes over, they integrate, and then you produce directly your therapeutic proteins in the vitreous and in the choroid, which is very important. Now, injecting in the ciliary muscle was not that easy. Electrotransfection was not that easy. And the idea, the IP there was to develop that specific device which you put on the eye, which guides you to do the injection exactly in the ciliary muscle and do the electrotransfection at the same time. The device which has been developed today, which is in clinic, needs to be optimized, like any device, unfortunately. So right now, it's being used in clinical trial, but it needs to be optimized. So talking about indication, so of course, she had to choose a target which was validated, because otherwise you don't know if it works, why, if it's because of a device, or if it doesn't work, if it's the device, is it the technology. So her decision was to choose non-infection uveitis and use the anti-TNF-alpha. Because we knew that anti-TNF-alpha works, Humira has been approved since, and that's the reason she chose non-infection uveitis first as a proof of concept to validate the technology, and that's why we're in clinical trial. I'm pleased to report today that we have 14 patients injected in Europe and in the United States, and after 6 months, we see a nice trend and efficacy signal. It's not enough to say that we have demonstrated a proof of efficacy, but what we're showing is after one injection, after 6 months, we see in this patient who have been already failed multiple previous treatment with non-infection uveitis, we see a decrease in the edema on the OCT, and we see an improvement in vision of more than 10 to 15 letters. And that effect is maintained for 6 to 8 months so far. So it's premature to say victory, but it's early enough to say, hey, we can make it. We can inject the plasmid. We can do electrotransfection. We can produce enough protein to have an efficacy level, and we can have enough durability for at least 6 months. So However, I'm sure you are all thinking, well, non-infection uveitis, this is the worst disease to choose. It's difficult to enroll patients, difficult to treat. So we all agree with all of you, and the investor will definitely agree with you. So what we are doing right now, we're focusing all our effort on two indications. One is retinitis pigmentosa, which is very close to my heart, as you suspect, after my 5 years at Foundation Fighting Blindness. And the other is the wet AMD. So why are we what are we doing for the RP program? For the RP program, obviously, we're not doing anti-TNF alpha. We have programmed the plasmid to produce human transferring. And what we're trying to do here is take any type of patient with RP, gene independent, and basically protect the photoreceptor from further dying. We know that in RP, your photoreceptors are dying slowly, and slowly the patient loses total vision and is totally blind. So the idea is to slow down the process of this photoreceptor dying with human transferring, and give extra years of vision to the patients. Did I express myself correctly?
Totally, and I get the concept on RP completely, and I want to come back to some of those other targets that you mentioned, RP, and I know how much you've been involved in that with the FFB. This is probably music to their ears, because I happen to know one of the board members. He's a member of Excite, and they were saddened by your departure, I'm sure, and they'll be glad to hear you're still plugging away at RP. And it makes a lot of sense as a target for this. It's a fascinating concept, but from the basics, you said, you take the DNA plasmid, if we were to inject it, quote unquote, in the traditional sense, it wouldn't make it. Why is that? Does it get enzymatically eaten up in the standard? What if you just loaded it in a needle and injected it into the ciliary body? What would occur in that situation?
Nothing is going to happen. What you do, you need to target your cells, and your DNA needs to penetrate in the cells. So if you do that, nothing is going to happen. It's the electrotransfection process which will allow the DNA to integrate. I'm not a cell biologist, so I would prefer Francine to respond to all this question. And I don't think we totally understand the process. But it's the same thing with gene therapy, except there you put everything in a virus with a promoter to target your cells. It's the same principle, except for us, we will not be permanent. We know that effect is going to last for 6 months, 8 months. We don't know yet. So you're probably going to have to come back with an injection. But I don't think that's a problem, because when we start to think about AMD, having the patient coming back every 6 months or 9 months for an injection or an administration, I think that's definitely music to the ear of people like you. And not having that virus always permanently here, well, you cannot stop the production of protein, is probably a plus as well.
It makes perfect sense. And I know that there are other groups who are very interested in trying to figure out ways to do gene therapy without the viral vector. This is brilliant. Is this then, most of us have no experience with electrotransfection, even conceptually, but this would then be a device that would be placed on the eye, which provides some sort of electric current that allows the biological material to penetrate through the sclera and into the ciliary muscle and into the cells.
So to make it simple, it's a little device. It's 1 centimeter long. It's not very big. It has a little guiding tool, because as you know, as a surgeon, I think injecting the ciliary muscle with just a needle is probably not that easy. So the curvature of that little device has been absolutely designed to mimic exactly the curvature of the eye, so you can penetrate on the side exactly in the ciliary muscle. That's the first thing, which I think is pretty brilliant. And then the electrodes come in, and they basically, by the current, by the pulse generator, they will, the device is linked to a generator. That little pulse will produce, will allow the plasmid to integrate inside the cells of the ciliary muscle. Francine would be probably much more better than me to explain.
Oh, that's very clear. And is this a device she developed, or is this something already available? She developed it specifically for this purpose.
Correct. And the IP is around that device. Absolutely.
I love the idea and did so right in the beginning when we talked with you and heard about Eyevensys some years ago, of the ciliary body as a target, which is something that I think has been unique to your company, to my knowledge. And using the term biofactory, which has been used before by other gene therapy companies, it seems to me the ciliary body is almost the perfect location and idea for that. Was that always the tissue target for her in this?
Yes, it was. And it was because for her, she knew it was safe. She knew from all her experiments, and we confirmed that from all the tox studies, that there is no tox issue. You can come multiple times. You still can produce the same amount of protein that was done in non-human primates, in rabbit, as well as in rat models. You can always come back multiple times. You can still produce your protein. You can do multiple injection. So it was safe. There was no issue with increase in IOP. There was no issue with inflammations. So for her, it was a very safe approach. And that was her motivation, her driver to go after that muscle.
It makes perfect sense. I think others will probably start thinking about that tissue because well, maybe not, but it's a great idea. The tumor necrosis factor itself, is this it's a cytokine? Is this a target that is currently in play in uveitis? I do treat uveitis. I don't know the mediators as well. Is this a mediator that is being approached currently with any systemic drugs, do you know, for treatment of uveitis?
And again, I'm not an expert in uveitis. Ron Bagage, with our CMO, was trained at the NIH in uveitis. Is better here. It's a complex disease. It's difficult to treat. Today, I believe you are all using Humira and you don't like it, right? Because you are not allowed to prescribe it.
Yeah.
But it needs to work. But what it does, I believe, it just it's a steroid-sparing approach, right?
Yeah.
And that's also one of the reasons we did not want to continue immediately with uveitis right now with this technology, because it's so difficult to enroll this patient and so difficult to treat. So that would need much more capital to continue. So that's the reason we're pushing right now on RP with the human transferring, which is basically trap the excess of iron. And then I'm going to start to talk about the IMG program, because for me, IMG is also music to my ear after all my years working on anicortavir acetate at Alcon.
Yes.
What is the problem today is injecting anti-VEGF. I think all you guys, you like it because it helps to get the patient some vision back. But at the same time, you're probably fed up to see all your patient every month in your practice, especially with COVID-19 right now. But the second problem, besides re-injection, is also fibrosis. And what we hear more and more, it's about the fibrosis issue. Well, what can we do with this plasmid? You can program them to produce two proteins. There is no cargo capacity issue like in a virus. In virus, you have cargo capacity issue. Here, you don't. So the idea, which I thought is really smart, is take the plasmid and try to program them to produce a aflibercept, an anti-VEGF, but also an antifibrotic, another protein. Combine them together, and here the hope is that what we hope to accomplish is prevent this multiple injection every month, but at the same time, try to prevent the fibrosis, because we know that after drying that retina with tons of anti-VEGF, you have this fibrosis produced and it doesn't help the patient. So that's the idea, and that's the reason we're trying to move also in this in the wet AMD indication.
Excellent. So that makes great sense with the anti-fibrotic strategy, and that would be wonderful, because that is kind of the rate limiting factor over and above the durability of the treatment. The fact that vision is still gradually lost from fibrosis is a problem. Where, more logistically then, and right up your alley as the chief, where is the company now with regard to these two programs? Where are you, and what are your next steps towards the RP and or wet AMD targets?
So we have two clinical trials going on for uveitis, which we are continuing in Europe and in the United States. But really what we're trying to do is really optimizing also the device. The device is its own component. I'm not totally concerned. If Sunil hears me on the phone, he probably would like, well, it's not that easy. We need to optimize the device. So that's why we're continuing the trials here in the Uveitis. The RP program, we plan to be in the clinic mid-year next year. Right now, we're doing all the IND enabling studies, planning to file an IND by midsummer next year. And then if everything is okay, we can start the clinical trial on RP probably September next year. The wet AMD program, we are right now able to produce the protein. We are in IND enabling phase. So we're going full blast, but we also have to do the non-human primate study because all the investors want to see non-human primate, even if it's not predictive. But we will have to evaluate our strategy here. Our goal is to be in the clinic more in 2022, early 23. That's what we try to do. So we try to accelerate all that as quickly as possible with money.
So money is always a limiting factor. I know this and you know this, and this is endless work for the CEO of any company, is raising the money, managing how much can be spent on various protocols. If you're moving forward, you're finishing your phase 1-2 trial now in uveitis, and let's presume the results are what you want to see and it's positive results, there's a good signal. Are you saying that you would then, in parallel, try to develop that into a phase 3 trial next, while then starting the protocols for the other conditions? Would you do those in parallel, or
no?
Let's assume you had the money to do so for a minute.
Well, yeah. Even if I do have the money, it doesn't grow on tree like.
Yeah.
So right now we're really putting all our effort on RP because we're very close. This is a program which is close, and on the wet AMD. The uveitis program, we're talking to other partners to eventually partner with other companies to develop the uveitis DME and maybe DME program. That's what we're thinking about. So probably do a partnership with another company for the 606, the anti-TNF alpha program. It's too much for a small company like us to do 3 programs at the same time. And uveitis is hard. It's very hard. So that's the reason we're going full blast after the RP program and full blast after the wet AMD program.
I've always been intrigued being in some of these discussions myself, but learning more about it over time. The position that you're in when you're presenting to potential larger investors as you move along in your program past proof of concept, say where you are now, how much push and pull do you feel as the CEO about the indications from some of these larger investors? How well do you have to draw a line in the sand, and how much do you have to acquiesce? Because you have a scientist on one side and an innovator who may have a certain idea about which direction. And then, of course, you may have somebody who wants to write a check for $20 million to help you fund a phase 2 or phase 3 protocol, who may have a different idea about indications. How often does that come up, and how do you manage these discussions?
So, as you know, it's money matters, and the investor, they want to see their money back. And so therefore they want us to push us in the big indication where there's a medical need. So today the big indication where there's a medical need is obviously wet AMD. We talked about durability, right?
Yes.
And making sure we have some tool that the doctors like you like to use. That's the reason I like the device. And the RP program, because it's hard. I mean, there's gene therapy, Spark was approved over there. And there is a better understanding that there is a huge need for RP and to find treatment for these patients. So I don't have too much pushback for RP and for wet AMD, because also RP, if you look at all the treatments which are being developed today, they are gene specific. And if you look at all the mutation for RP, I think there are about only 10 to 15% of the gene mutation which are touched today while treatment being developed for XLRP or RP65, etc. So not too much pushback. Uveitis, I have pushback. I'm not going to lie to you. I have pushback. One of the reasons, difficult disease, orphan disease, difficult to enroll patients, and even if you have a data, difficult to interpret. very difficult to get money for uveitis.
You're not the first person who I've seen to experience that, even recently. And it's kind of tough because as a clinician, we know how challenging this disease is, and we would love for our patients to have options beyond steroids and some of the current chemotherapy. But I totally understand. I'm also an investor. As an investor, why the bigger investors certainly are pushing you always towards to the big C markets, wet AMD and such things. RP is a little bit in between, and I'm glad to see that you're approaching that. It's an area of expertise for you. Would this be somewhat agnostic to the type of RP, or would it be very specific, again, like some of these one gene type diseases?
No, and after my 5 years at Foundation, that's what I like about our RP program, because first it's gene independent.
Yeah.
And honestly, if it would be my son, having somebody messing around under my retina and injecting a bleb, and I'm not sure how long it's going to last, it's going to work, I don't like that. So at least I believe with our approach, we can give a chance to slow down the disease. We're not going to cure it, but we can slow down the disease. And I think this is used for this patient, especially for these kids. And in addition to that, while gene therapy continues and somebody's going to work maybe on cell therapy, nothing prevents you to combine both together. Nothing, because when you have gene therapy and you do the injection of a bleb, the disease still continues. So if you think about our approach, we don't have a virus. So you can think about, you give our treatment, and if you want to do gene therapy to your patient, because in between you have more information about his gene mutation, and Ola has a new gene therapy treatment which is developed for specific for that mutation, they can go together. One doesn't prevent the other. And I think that's we have value with our approach as well there. Does that make sense?
It totally makes sense, and I think it's correct. One final question or thought, and I'd like your thoughts on it more broadly. Non-viral gene therapy, that's kind of the theme of this company and this discussion, and it's fascinating, and I think it'll be a new concept for a lot of people who might listen to this. Do you think ultimately that is going to be the method, not necessarily this exact method with this indication, but overall, do you think gene therapy in the eye, let's say, will ultimately be more realistic down the road with a non-viral technique?
I think it depends on the indication. If we talk about AMD, I mean, I'm not convinced that having a gene therapy that you produce non-stop with anti-VEGF, it's good for the retina. I mean, you are the expert more than me, but I'm not going to believe that. The problem with gene therapy, if you target a disease like wet AMD, you're going to produce and produce and produce this protein non-stop. For a gene mutation like in RP65, for example, totally different discussion. I get it. I understand. If you can correct it permanently with mutation, yes, there's definitely value for gene therapy. But for wet AMD, I don't know. I don't know.
No, your point's well taken. There, we definitely have the sense that overdoing it in busting down VEGF has its own potential cadre of problems. So your point's well taken, and a lot of people are talking about this. And how do you turn it off if you had to turn it off, or how do you fine tune the dosing in such a paradigm? So I think your point's well taken, and a lot of smart people are talking about it, including you. Thank you for coming on. I really appreciate learning about this. This is very unique stuff, and I know about your company from before, and I'm congratulating you, and I'm wishing you well. I think this is a technology that would be wonderful for the eye business.
Well, thank you so much for having me. I appreciate, and credit goes also to Francine.
Yes, no, I know, and I please thank her for allowing you to give us this information, and she's welcome to join in at one point if she wants and elaborate. We'll have you both back at some point.
Thank you so much.
Thank you very much.
Thanks for joining us this week. And don't forget to sign up for the OIS Retina Showcase, which will feature exciting retina startups presenting fresh clinical data and will be followed by a panel of clinical key opinion leaders. Sign up today at OIS.net.